Welcome to Creating Your Genius.
Artlist.io.
This episode is going to focus on energy, metabolism, inflammation, and a whole host of other important biochemical reactions. Prepping for this episode has been a lot of fun because I really enjoyed biochem in my undergrad. This episode has been a chance for me to revisit a lot of the topics that I found so fascinating because they relate so closely to the world of epigenetics, and it's just very important to understand the biochemistry of what's going on in the body. You could just take my word if I made a statement like being exposed to mold disrupts how you produce energy. That statement on its own might mean something to you, but it might not mean anything. But if you know how we produce energy, some of the genes and proteins that may be activated during an immune response, that statement then carries a lot more validity to it because you understand a little bit more about why exactly mold would be disrupting our energy production. In the context of epigenetics, it's really important because this biochemistry is really the link between the environment and the genes. There are so many chemicals that we are exposed to in the environment, not just things like toxins, but also food is chemicals, water is a chemical, sunlight could technically be classified as a chemical. And so if we're introducing chemicals into our biology, that, for lack of a better term, is biochemistry. So let's get right into it. First, what is energy? Why is it important? Each of our cells has quite a bit of work to do every second of every day. Whether we're awake or asleep, the cells are constantly sensing the environment, activating genes, creating proteins, dividing, reproducing, carrying metals, transporting molecules throughout their own cells or in between other cells. And there's a whole other list of functions. So in order to carry out these life-giving tasks, they need energy. So when we start with energy, it's really important to know the first law of thermodynamics, which says energy cannot be created or destroyed. So that means when we're creating cellular energy, it's actually just a transfer from one source to another. And the transferring of the energy is what powers our cells. If we're not able to create energy from nothing, we have to get it from somewhere so that we can transfer it through all sorts of different reactions. And where we get the stuff is food, water, oxygen. And sunlight is important for energy production. It doesn't carry the same weight as those other three because we're not able to use the ultraviolet radiation and turn it directly into energy like plants can. We digest the food, and as we break down the large piece of food into smaller molecules, energy is moved, and that's how we power our cells. That's a pretty gross oversimplification, but as we dive deeper, this transfer of energy is done mainly through the transfer of electrons and hydrogen atoms. Hydrogen's a really important atom and element because it's the smallest element on the periodic table and it only carries one electron in its outer shell. So when a hydrogen is transferred from one molecule to another, in essence, one electron is moved. And as the reactions happen over and over again in our cells, it really is sort of just the transfer of hydrogens and electrons. The sum of all of these reactions that happen in our body to produce energy is what we call metabolism. And metabolism can be split into two subcategories of reactions. They are either anabolic, which is building things, or catabolic, where things are broken down. Digesting food is a really good example of catabolism, and building bone and muscle is an example of an anabolic reaction. Anabolic is a term that people often throw around with steroids, and rightly so, because they are compounds that we use to help people build, and specifically build muscle. There's always this balance of breaking down and building up, because food isn't the only thing we break down. Sometimes we have to break down our own proteins for energy. We also have to break down and digest proteins that have been damaged or aren't working correctly anymore. And ideally, there's always this balance of breaking down the right things and also building the right things. When the building gets out of control, you can have things like cancer, which is, for lack of a better description, when cells that are supposed to die don't die, and they aren't broken down in a catabolic reaction. They just continue to grow and build, and that growing gets out of control, and it causes dysfunction. All of these reactions are mediated by proteins, which are coded for by genes. Go back to episode 1 for a little more in-depth information about that. And these specific proteins involved in facilitating chemical reactions are what we call enzymes. The presence of enzymes make the reactions proceed extremely efficiently. We wouldn't be able to break down any of our food without digestive enzymes, and then we wouldn't be able to convert the broken down food into energy without other sets of enzymes. So we have the enzymes that act on the targets, which is called a substrate, but a lot of times these enzymes, they can't work just on their own. They need something called cofactors to allow them to work properly. And so these cofactors are molecules that are non-proteins that are required for the proper enzyme function. And common examples of cofactors are vitamins, minerals, metals, and something that we call essential nutrients, which are things that are necessary for enzyme function, but don't really fit into those other groups. Getting sufficient cofactors is why eating healthy foods and nutrient-dense foods is so important. We can have all the macronutrients that we need, proteins, carbs, and fats, but with no cofactors, it's going to be very hard for our body to break them down efficiently, because the enzymes that help break them down, they're not going to be working. There are different dietary sources for all these different cofactors. For example, vitamin C is mainly found in plants. Oranges or citrus fruits are a famous example of rich vitamin C foods. B vitamins are abundantly found in animal protein sources. Vitamin A is almost exclusively found in organ meats. Vitamin D can be found in fish oil as well as organ meats. Iron is abundant in nuts. Potassium is a mineral, which is found in bananas. And then an example of an essential nutrient is something called choline, which helps us break down our fats. And this is found abundantly in eggs, and is one of the reasons why really delicious and good egg yolks have that dark orange color, as opposed to being that light yellow. When the enzymes have sufficient levels of their cofactors to work, they modify their target, which is the substrate that I mentioned earlier, and they turn it into something else. And then that something else is further modified by another enzyme that most likely needs a different set of cofactors. Taking a step back, because enzymes are coded for by genes, it's possible that variation in the genes can affect the function of the enzyme created. Sometimes this leads to an enzyme that doesn't work very well, and other times it can actually cause enzymes that are more active. This genetic variation happened over generations and generations as we evolved, and it is likely due to people living in different parts of the world where different cofactors were more abundant than others. These genetic variants can go a long way in explaining why some people feel fantastic when taking certain vitamins and minerals, where others don't feel much of a difference. The people who felt a big difference likely had genetic variants that caused the function of the enzymes to be less than optimal. And when they supplemented with the cofactors, it helped boost the function of that enzyme. Taking that little pit stop and going into enzymes and cofactors is really important in the context of creating energy, because there are so many reactions and so many steps that go into converting our food into energy, and it requires a whole host of enzymes that all need specific cofactors. What is our cellular energy? This is something called ATP, which stands for adenosine triphosphate and is known as the currency of energy in our cells. The structure of ATP is one adenosine and three phosphate groups, which are made up of oxygen and phosphorus, and the bonds between these groups are very high energy. So when these bonds are transferred from ATP to other molecules, a lot of energy is moved. The process of breaking a phosphate bond in ATP and donating that phosphate group to another molecule is something called hydrolysis, which involves adding a water molecule to the ATP, which helps facilitate the breaking of the phosphate bond and the transfer of the phosphate group to the other molecule. This is one of the reasons why staying hydrated is so important, because we need water to move energy. And if you don't have enough water, energy is not going to be moved quite as efficiently. When this ATP transfers its phosphate group and energy, it becomes known as ADP, which still contains the adenosine, but with only two phosphate groups attached to it. And when we get into inflammation and how the cell senses energy, this ADP becomes very important. Where is ATP produced in the cell? This is going to take us back to 7th grade biology, and it seems like the only thing people remember from that class is that the mitochondria is the powerhouse of the cell. And that's because the mitochondria is where almost all of the ATP is made, using a specific enzyme called ATP synthase. These mitochondria are fascinating organisms because they are ancient and they had an origin outside of being in human, plant, and animal cells. We co-evolved with them, and they have their own genome. There are these little life forms living in our cells that help us create energy. It's really kind of a beautiful process. We start the energy process by consuming food, breathing, and drinking water. It seems like there's a long way to get from eating a potato to our mitochondria creating ATP. And that's true. There are different processes that the body uses to break down fats, carbohydrates, and proteins to make energy. A quick little introduction to these molecules. So carbohydrates are made of carbons, oxygens, and hydrogens. Carbon, hydrate would be water. So it has carbons, oxygens, hydrogens. Those oxygens and hydrogens is water. Fats are different from carbohydrates as they are made of just carbons and hydrogens. Proteins and amino acids are going to be the most complex out of these three, and they have carbon backbones. They contain oxygen and hydrogen, but they also contain nitrogen. And because of that nitrogen, using proteins and amino acids to create energy can be very metabolically expensive, and ideally is not used. And the term metabolically expensive means you're breaking down more than you're building. I'm going to mainly focus on fats and carbs as energy sources because ideally that's what we're using. So we'll start with carbs. Breaking down carbohydrates into energy requires a few different steps. The first step is a process known as glycolysis, where after you break down a carb, we'll use the example of a potato. You break down that potato into glucose molecules, and glucose is shuttled into glycolysis. A really important enzyme for glycolysis is something called phosphofructokinase, or PFK. And this step controls whether or not the cell commits to glycolysis. Once PFK is activated and the glucose is modified, the cell is committed to glycolysis, and it goes through this process to create a molecule called pyruvate. Pyruvate is then moved into something called the citric acid cycle in the mitochondria. An important distinction between glycolysis and the citric acid cycle is that glycolysis happens in the cytosol of the cell, meaning it's outside of the mitochondria. So in order to get this pyruvate into the citric acid cycle, it has to be transported through the membranes of the mitochondria. Pyruvate is then put through the citric acid cycle. During this cycle, as the pyruvate is modified, there are different transfers of hydrogens and electrons to something called electron carriers. And those carriers take the electrons and hydrogens and they move them to something called the electron transport chain, where those electrons and hydrogens are used to power the ATP synthase that puts a phosphate group on ADP to create ATP. I understand that's a little bit complex, but the main takeaway is that we've got the citric acid cycle, which donates electrons to electron carriers, and these electron carriers move the hydrogens and electrons so that they can be used by ATP synthase to create ATP in the mitochondria. So an interesting point for glycolysis is that during the breakdown of one glucose molecule, you actually create one ATP in the cytosol, which is readily available to be used for energy. When ATP is made in the mitochondria, it has to be transported from the mitochondria into the cytosol to then be used. This factor of glycolysis is going to be really important as we get into inflammation. Before I move on, I want to go a little bit deeper into the electron carriers. These are molecules known as NAD+ and FADH. When electrons in the form of hydrogens are donated to these carriers, they are reduced to NADH and FADH2. And these are vital for our ability to produce energy because they facilitate the transfer of electrons to power ATP synthase. These electron carriers donate their electrons to the electron transport chain. And this electron transport chain takes the electrons, but it actually pumps the hydrogens through the mitochondrial membrane. So it creates a situation where there's a lot of hydrogens on one side of the membrane and not many on the other. This difference of concentrations of hydrogen is one of the factors that powers ATP synthase. So there's the flow of electrons through the electron transport chain, as well as this creation of a hydrogen gradient. And the combination of those two things is moving a ton of energy. Specifically, the difference between the concentration of hydrogens on one side of the membrane and the other cause the hydrogens to want to flow from where there's a high concentration of them to a low concentration. And this movement of hydrogens is a lot of energy in itself. And it's enough energy to allow ATP synthase to make massive numbers of ATP. This process of using the hydrogen gradient and the electron transport chain to create ATP is called oxidative phosphorylation. The combination of glycolysis, the citric acid cycle, and oxidative phosphorylation is what goes into breaking down carbohydrates into energy. The process for breaking down fats is going to be a little bit different. When you break down a fat, oxidative phosphorylation and the citric acid cycle are still used, but glycolysis is not, because glucose is the required molecule for glycolysis. When we use fats for energy, the fat cells are transported to the liver, where they're broken down through a process called beta oxidation. Because fats are made of only carbons and hydrogens, there's the potential for the movement of quite a few hydrogens. A key difference between carbohydrates and fats comes from the number of carbons in the chain of the molecule. For glucose, one of the most common sugars that we use for energy, and fructose, another one, there are 6 carbons in the backbone. But for a fatty acid, there could be up to 20 carbons in the backbone of the molecule. And so each time beta oxidation is run, the end product takes 2 carbons off of that chain and removes 2 hydrogens to give to NAD plus and 2 hydrogens to FAD. And those electron carriers go to the mitochondria and donate their electrons to the electron transport chain. So because fats have up to 20 carbons in there, there's the potential to make massive amounts of NADH and FADH2, which then go and donate their electrons to the electron transport chain, which through oxidative phosphorylation makes a lot of ATP. The potential to create so much ATP is why fats are known as the most energy-dense molecule, and why we use them to store energy for when things get a little bit difficult in our environment. So for one 18-carbon fat molecule, we have the potential to make over 100 molecules of ATP. Compare that to glycolysis. For one glucose molecule, you can make just about 33 ATP. So there's potential for fats to create much more ATP than through breaking down sugars. So just a few key differences between glycolysis and beta oxidation are that glycolysis is able to directly create ATP, where beta oxidation does not, and the molecule that's introduced into the citric acid cycle is going to be different. So for glycolysis, it's pyruvate, and for beta oxidation, it's a molecule called acetyl-CoA, which is able to be incorporated into the citric acid cycle to create the NADH and FADH2 that are then used in the mitochondria to make energy. As our cells are making all of this energy in the form of ATP, there's got to be a way for them to sense what's going on, whether we have enough energy, whether they need to upregulate oxidative phosphorylation, glycolysis, beta oxidation, and other processes to allow the cell to continue functioning. So the cell is always sensing the ratio of ATP to ADP. ATP is readily available energy that the cell can use to carry out functions. ADP, because that phosphate group has been donated, is no longer able to function as an energy carrier. It needs to be recycled back into ATP. The more ATP in the cell, the more energy. And high levels of ATP inhibit glycolysis and inhibit the citric acid cycle. But when ADP levels start to creep up, the cell senses danger, because that is the signal that there is low energy in the cell. Because these levels of ATP are so tightly regulated, when there's disruption in this and ADP levels start to rise, the cell is going to prioritize making quick, readily usable energy over making the absolute most amount of energy that it possibly can. As a compensation mechanism, when ADP goes high and ATP starts to drop, the cell upregulates glycolysis, because glycolysis makes that one readily usable ATP per glucose molecule that's broken down. Ideally, because ATP and ADP are always being exchanged in the mitochondrial membrane, the ADP is going to be transported into the membrane, turned back into ATP, and then shuttled back out into the cell for use. So the recycling of the ADP is going to be the most efficient form of creating energy, and that's through oxidative phosphorylation and the citric acid cycle. When this process is happening correctly, the cell doesn't really have to run glycolysis a whole lot. Creating energy efficiently is pretty fantastic, but obviously there can be factors that can mess with this energy production. And this is why the topic of inflammation ties so tightly to energy production, because the biggest reason that ATP levels drop in the cell and ADP levels rise is going to be inflammation. Inflammation is a word that's thrown around quite a bit. And simply put, inflammation is the body's response to some sort of insult. These insults could be infections from bacteria or viruses. The insult could be from a food that you eat. Like an allergic reaction is a great example of an insult via food. These insults could be food additives, they could be environmental toxins, they could be high blood sugar, they could be high levels of free radicals. There are many different things that can go into causing an inflammatory response in the cell. And the inflammation happens when there's something that the body sees as a threat and needs to use resources to deal with. So inflammation is definitely not a bad thing. Actually, if we didn't have inflammation, we would all die pretty quick and horrible deaths, because we wouldn't be able to kill pathogens, we wouldn't be able to recover from strenuous exercise, the bacteria in our gut would completely overgrow and kill us. So we need inflammation. When these insults happen, the immune system is activated, and the activation of the immune system is what's going to cause the inflammation. That's where you get your pain, your swelling, redness, heat. That's known as acute inflammation, and that usually calms down once the immediate stimulus is dealt with. So think about, when you get a cold. You get exposed to the virus, you're fine for a couple of days, then you get a fever, body aches, you're sweating, face is all red, and you have a fever for 24 hours, 48 hours. You have that fever until you kill the pathogen, and then once your body no longer senses that threat, there's no need for the inflammation anymore. The inflammation is then calmed down by processes in the body that are immunoregulatory, and everything's great. That's acute inflammation. What can happen is something called chronic inflammation, which basically is when the threat is never really removed, or if the threat is coming from a process in the body that goes haywire. Something like high blood sugar is a process in the body that's happening that kind of gets out of whack, that causes chronic inflammation. And this is part of the reason why diabetes is such a serious condition, because when you don't have the insulin to deal with the blood sugar levels, they stay circulating and they can get oxidized and create free radicals. And these free radicals are going to stimulate the immune system, and it can cause a cascade that is a lot of pain and all sorts of dysfunction. Chronic inflammation has been linked to several different types of cancers, autoimmune diseases, type 2 diabetes, which I mentioned, cardiovascular diseases, and basically all diseases of aging have an inflammatory component to them. So when the immune system's activated and this inflammatory response happens, it disrupts our normal metabolic function. During the immune response, there are molecules called cytokines which are produced that tell the body that it's time to fight. The immune response takes a lot of energy, and we need this energy very quickly. And because of this urgent need, the body is going to switch up the way that ATP is produced, and it's going to upregulate glycolysis, because glycolysis makes ATP without the energetic cost of transporting it out of the mitochondria. When glycolysis is upregulated, it's actually going to decrease mitochondrial function, meaning that there's going to be less overall energy produced. When the body senses a threat that it needs to take care of quickly, this is fantastic. But during chronic inflammation, there's going to be chronic downregulation of mitochondrial function, which over time is going to seriously deplete how much energy that your cells can make. One of the reasons why there's so much fatigue during inflammation, both acute and chronic, is because the total body system's energy expenditure can be up to 10% higher. And the body makes up for this by, again, upregulating glycolysis. And so you have a short-term boost, but long-term, your body's running faster and burning up more energy, and you're not able to keep up with the demand, which is going to lead to fatigue. So, big picture here. The body's exposed to an insult. The immune system is activated as the receptors on immune cells sense the environmental change, which could be high blood sugar, a pathogen, a virus, or all sorts of things. The immune cells are activated. They release cytokines that coordinate the immune response to deal with the threat correctly. These cytokines are proteins, which are encoded for by genes, and they're activated by environmental factors that cause the cytokine genes to be upregulated for the response. And these cytokines can have wide-ranging effects in the body. So there are three that I want to focus on: IL-6, IL-1, and TNF-alpha. All these are encoded for by genes with those same names, so IL-6, IL-1, TNF-alpha, and they're all inflammatory, and they're activated during the immune response. IL-6 is really important during the acute phase of the immune response, and it helps immune cells develop into antibody-producing cells. And antibodies help us clear pathogens very efficiently. Its role is recognizing and coordinating the immune system to deal with the specific threat. IL-6 has been shown to cause dysregulation in the division and replication of mitochondria. And this is important because when cells have been infected, it's very important that those infected cells aren't allowed to divide and replicate, and you want the cells that are infected to have the least amount of energy possible. Part of what happens is these cytokines are toxic to the mitochondria, and when the mitochondria of the infected cells don't work, they can't replicate and they die off. The next cytokine is TNF-alpha, encoded by the gene TNF-alpha. It's also very important for the killing of pathogens and the induction of inflammation. And TNF-alpha exposure to neurons cause their mitochondria to reduce respiration, which is the production of ATP. IL-1 plays a role in the formation of cellular machinery that drives inflammation and is specifically implicated in the development of fever. IL-1 is very interesting because it's been shown that high levels of IL-1 in fat cells decreases mitochondrial oxidative function, the ability to make ATP. And high blood sugar was shown to stimulate IL-1 in mice. I think you're noticing a common theme here, is that these inflammatory cytokines are pretty toxic to the mitochondria. And it would follow that long-term exposure of our cells to high levels of these cytokines is going to mean that we aren't producing energy very efficiently, and we're going to feel pretty crappy. So what exactly can cause these inflammatory cytokines to be high levels? I mentioned before that it's when the stimulus is not removed. But what does that mean? Well, that could mean because the stimulus for your inflammation is something that you eat all the time, like fried food, for example. Or it could be something that you live in and you're breathing in all the time, like mold. It could be from underlying infections that are able to hide, like Epstein-Barr, or it could be from genetic variants that cause overactivation of the cytokine proteins. And all of these things can lead to chronic inflammation. Especially when there are variants, the immune system becomes really trigger happy, especially in regards to IL-6 and TNF-alpha variants. So the immune system is almost too good at detecting threats, and it keeps the inflammatory response too long and makes it too strong. So it's not able to calm itself down very well. And so there's a lot of strain. There's going to be a lot of hijacking of energy, increased glycolysis. The mitochondria are going to be downregulated, and there's going to be lower ATP. All right, so what do we do about this? The easy way to say this is find the inflammatory stimulus and remove it. You change the environmental inputs, and that changes what the cells are sensing, and that changes the gene expression. So if you're breathing in mold all the time, your body senses the mold, TNF-alpha and IL-6 are upregulated because the mold is a danger, it's toxic to the cells, and those genes are activated because really it's the correct response. So if you're able to get out of the environment, your cells are no longer sensing that toxin, and ideally they would be able to regulate themselves, decrease the immune response, and things go back to normal. But oftentimes there's combinations of these inflammatory stimulus. You've got mold exposure and IL-6 or TNF-alpha variants. And so changing these inputs doesn't have to be just getting out of the environment. It can be giving something that's been shown to downregulate cytokine genes and activate the proteins. So things like antioxidants are good examples of compounds that are used to help decrease inflammation. And part of the way that it does this is by downregulating cytokine genes and proteins. A pretty famous one is curcumin from turmeric. And curcumin has been shown in multiple studies to downregulate IL-6. And there's one study that actually proposed that curcumin's anti-inflammatory effects are actually directly from this downregulation of IL-6. So if you're struggling with inflammation, if you've got pain, curcumin could potentially be a great option. Glutathione and N-acetylcysteine, which is a precursor to glutathione, are two examples of antioxidants that neutralize free radicals. They've been shown in studies to help with pain and to lower inflammation. So those are two ways that you could support your genes and change your environmental inputs. And this one's not fun, but one of the most common inflammatory stimulus is food. I mean, it's fun to talk about, but in practice, it can be very hard to get rid of inflammatory foods because they seem to be everywhere. One of the most common examples is fried foods. Fried foods contain something called peroxidized lipids. Basically, these fats that we fry foods in, when they're exposed to high temperatures and high concentrations of oxygen, which happens when you fry them, the oxygens get turned into free radicals that attack these fats. When the free radicals attack the fats, it basically turns those fats into free radicals. And there's the potential for pretty wide-ranging inflammatory effects from these peroxidized lipids. A study showed that in mice, when they were exposed to these peroxidized lipids, levels of IL-6 and TNF-alpha were shown to increase. They were having an immune response to the food that they were being fed. Obviously, we're a little different than rats, but they're living organisms just like we are. And if they're responding in an inflammatory way to something, wouldn't it be possible that we are as well? It's not to say that 100% yes or no, but there's a reason why we use rats to study things, and that's because a lot of times they're pretty good analogs for what would happen in us. Removing foods like fried foods, that's another change in your environment. and you're going to down regulate the immune response by not exposing yourself to that stuff. And just like when we were talking about acute versus chronic inflammation, the levels of cytokines and gene activation for chronic exposure to these peroxidized lipids is going to be different. So during the acute response, the TNF-alpha level raised significantly in rats, and the IL-6 was raised moderately. But as these rats were continually exposed to the peroxidized lipids, the TNF dropped, still stayed elevated, and the IL-6 raised extremely high. So this is to say that the length of the stimulus is going to matter in how your genes are expressed. It's not going to be TNF-alpha goes through the roof all the time, whether you eat fried food one day or 30 days in a row. It's that immune response is going to be regulated based on how the body sees the threat. And in these rats, it was shown that the TNF-alpha was the first one to go up, and the IL-6 was raised later. And like I mentioned when I first introduced these molecules, all three of them, TNF-alpha, IL-6, IL-1, have been shown to have direct impact on our mitochondrial health. So that's could be one of the reasons why after I eat fried food, I know, I feel like crap. I'm tired, I can get achy, and I can feel brain foggy. And it's probably because I'm mounting an immune response. My body's like, all right, we got to mobilize some resources to deal with these toxins. And, once in a while, it's all good. I'll wake up the next morning feeling like crap. But then as that day goes on and as, I take care of that toxin, I bounce back really quickly. But if you're having it over and over and over again, months, years, we just don't know what types of chronic issues that could cause. There's a lot of resources out there to help people with diets to adjust them for inflammation. There's stuff like food inflammation tests. There are a bunch of brilliant functional medicine doctors, health coaches, and dietitians that can help. And modifying your diet is definitely something you can do on your own. Obviously, it's important to make sure you're getting enough of the macronutrients and vitamins, minerals, cofactors, essential nutrients, but trying something like eliminating fried foods for 2 weeks isn't going to impact the levels of vitamin A you're getting. If anything, as you replace the fried food with something probably healthier, you might get more of these essential nutrients. One of the things that I always tell my clients when I'm working with them is the body's really smart and it adjusts pretty quickly to diet changes especially. You'll know if something's working for you, especially if you make a drastic change like going to a vegan or a carnivore or keto or some of the more restrictive diets out there. Your body's going to give you pretty quick feedback. You'll know within a couple weeks whether this is something that's good for you or whether your body's like, hey, let's take it easy here. So if you are going to try a new diet, listen to your body, and there's many resources out there that can help you. It doesn't have to be super daunting. There are some really easy ways, like eliminating fried foods or taking a little bit of curcumin, that you can change the inputs and change that gene expression to help you feel a little better. I know this episode was quite a bit of biochemistry. I find it fascinating, but I realize that not everyone loves it quite as much as I do. But knowing how your body changes when you're producing energy based on carbs or fats, and how you produce energy differently when you're under an immune response, and some of the things that can cause an immune response that you might not be thinking of, really highlights that the biochemistry is so important. It's the link between what's going on in our environment and what's going on in our genes. I hope this information helps empower you to take some of your genetics into your own hands, downregulate that TNF alpha, downregulate that IL-6, and to continue to create your genius. And thank you for tuning in to the first full-length episode. I will see you next time.